Hi folks, so now we're going to talk about B cell maturation. So all the steps that are involved in developing a mature antibody producing B cell. But of course there's a range of steps that a B cell has to go through to get to the stage where it's producing antibody. Okay, so to start with, the B cell will mature in the bone. Then it will move to the spleen or other lymph nodes such as tonsils as well, and undergoes a process of maturation in those areas. And once it's went through that stage of maturation, it will then make its way to lymph nodes where it will await antigen. And when the B cell receives antigen, it will receive help from a T cell and subsequently go through the processes of producing antibody against the antigen. That's a very brief summary. So it's in the bone that the B cell will start out as a pre-B cell. There are a series of stages of that process where pre-B cells undergo B cell receptor rearrangement. Okay, folks, just to reiterate that all immature B cells start off with the same B cell receptor, and this initial stage of maturation involves that B cell manipulating its B cell receptor genes so that that specific B cell has a unique receptor. Okay, as I said before, a mature B cell, each individual mature B cell will have its own unique version of that gene. Okay, so that's what happens during the pre-B cell stages of maturation. It undergoes that process of B cell receptor gene rearrangement. Now, at the end of that the stages of that process, it then becomes an immature B cell. Okay, so an immature B cell will leave the bone marrow and make its way towards other lymph organs such as the spleen and the tonsils. In order for the pre-B cell to become an immature B cell, it needs to go through the stages of producing a functional B cell receptor gene. Okay, so during the initial stages, it rearranges its gene, and in order to proceed through the process of maturation, it needs to test its B cell receptor, its new B cell receptor, to make sure that it works. And it also needs to go through a series of stages where it tests the new B cell receptor to ensure that it doesn't react to self-antigen. And that stage is called negative selection, which we'll cover in a little bit more detail. But once it goes through that stage of A, checking its B cell receptor works, and B, making sure it doesn't react against self-antigen, at the end of that process, the immature B cell is now ready to leave the bone. Once it leaves the bone, it migrates towards other lymphatic organs such as the spleen or the tonsils. And when it arrives, in particular when it arrives at the spleen, it becomes a transitionary B cell. So the B cell arriving at the spleen from the bone then becomes a transitionary B cell, and during that process, the transitionary B cell goes through stages of maturation, where firstly it undergoes a further process to make sure that these transitionary B cells don't react against self-antigen. And the last stage of the transitionary B cell test is to ensure that it does react against its target antigen. Okay. So once it goes through that transitionary B cell stage, it's now ready to leave the spleen, or tonsils as it may be, and it then subsequently makes its way towards a lymph node. And when it arrives at the lymph node, it simply awaits for the antigen to arrive, and when the antigen arrives, whether it's a pathogen or otherwise, it will receive help from a T cell and subsequently then go through the stages of producing antibody. So that's just a very quick synopsis of the stages that we're going to talk about. Okay, so you go from a pre-B cell, make sure that the B cell receptor gene is rearranged. Then we make sure the B cell receptor gene rearrangement has led to the reduction of a functional receptor. We then check that that receptor doesn't respond against self-antigen. And then at the end of that process, this immature B cell makes its way from the bone to the spleen. Upon arriving at the spleen, it becomes a transitionary B cell where it undergoes a series of checks to make sure it doesn't respond against self-antigen again, and it also undergoes a check to make sure that it can respond against the antigen that it's targeted against. Once that process is over, it then leaves the spleen, goes to the lymph node, and in the lymph node awaits stimulation by the antigen of the pathogen. Okay, so now to go into a little bit more detail. So in the bone, the B cell undergoes immunoglobulin gene rearrangement. So that'll be the heavy chain and the light chain genes will undergo rearrangement. And if that process of rearrangement is successful, then the B cell will get a signal to survive. And that process is called positive selection. Okay? So once the B cell produces a functional B cell receptor, it will get a signal to survive. Of course, if it doesn't produce a functional B cell receptor, a number of things can happen. One is that it will try to fix that gene and hopefully produce a functional B cell receptor, or it simply won't get the signal to survive. Another stage is when the B cell receptor is tested to ensure that it doesn't react against B cells. That it doesn't react against self-antigen. Okay? So this stage is called negative selection. And essentially what that involves is the B cell receptor is expressed on the surface of the cell, and it's tested to see if it reacts against self-antigen. If it does, there's a number of things that we can do in this negative selection process. We can eliminate that cell because it does react against self-antigen. We could do that. But we could also disable the B cell. So we could disable the B cell so that even if it does get stimulated, it won't have the ability to elicit a strong antibody response. Okay, and that would be called anergy, which we'll cover in a little bit of time. The other thing we can do is, because this B cell does react against self-antigen, we have a last minute ability to change the B cell receptor gene again. And the aim here is if we can amend the B cell receptor genes, then if we amend it in a way that it no longer becomes self-reactive, then it can then proceed to become a mature B cell. Okay? So there are the stages that happen in the bone. Positive selection, i.e. make sure you produce a B cell receptor that works. A negative selection, make sure you don't produce a B cell receptor that will react against self-antigen. So at the end of that process, we then have an immature B cell ready to leave the bone marrow. And this immature B cell, the majority of them won't react against self-antigen. But we'll cover that in a little bit more detail because there are a subpopulation of B cells that manage to escape this negative selection. Now, whilst there are a number of B cells that can escape that negative selection and still respond against self-antigen, the immune system has this security net that when the immature B cell leaves the bone, it then arrives at the spleen. And of course, there's some evidence to say that it can also arrive at the tonsils for this next stage. So what happens is the B cells arrive in the spleen and they automatically become transitionary B cells. And the whole concept of these transitionary B cells is that they get a further check, again, as I said, a security blanket, if you like, for the immune system, that we undergo another process of negative selection to make sure that these B cells do not have a B cell receptor that reacts against self-antigen. Okay? And after it also gets checked to make sure that the B cell receptor can respond against the antigen that we want it to respond, the pathogenic antigen, then it can leave the spleen as a mature B cell, a naive mature B cell. Naive simply means that it hasn't been stimulated by the antigen at this point. Okay. So these are the steps that are involved in going from a pre-B cell to an immature B cell. Okay. First of all, you have the stem cell, the hematopoietic stem cell, that has the ability to self-renew. And what happens then is it then receives a series of growth factors that causes it to differentiate into a pro-B cell. That pro-B cell then undergoes gene rearrangement, and we'll cover this in a bit more detail, but undergoes gene rearrangement in the heavy chain, and it subsequently matures to a large pre-B cell, where it undergoes further amendments to the heavy chain, and then once that's finished, it then undergoes a series of proliferation, and they then differentiate into a small pre-B cell where the light chain of the antibody or the immunoglobulin will be rearranged. And once that's completed, then the B cell will develop into an immature B cell, and it'll be checked to make sure that its B cell receptor is functional and it's not self-reactive. Okay? And once we're at that stage, we have an immature B cell that has a functional receptor that will not react against self-antigen. Okay? Now, we've now moved from a pre-B cell to a immature B cell that's now going to make its way towards the spleen. Okay, now in terms of the series of steps that are involved for negative selection, that is, when we do get a B cell that reacts against self-antigen, we have a series of options which are nicely highlighted in this slide. So if you produce a self-reactive B cell, which you can see on the left-hand side, you have the ability to functionally silence that B cell. Okay? And what happens basically is that it causes a cell signal within that cell to ensure that that cell will not respond, it will not create an immune response if it's stimulated by the antigen. So that B cell can simply leave the bone marrow or the spleen, despite the fact that it reacts against self-antigen, and the security for the immune system here is that if it does get stimulated by that self-antigen, it won't create an immune response. And that concept is called clonal energy. It's essentially, it doesn't kill the B cell, it just makes the B cell ineffective. And it makes it susceptible to T cell death. Okay? So that B cell could go out into the systemic body, meet a corresponding T cell, and that T cell will kill it. Another option is that if the B cell reacts against self-antigen, it will induce cell death immediately. Okay, so if we have a self-reactive B cell, the immune system has the option to just kill it and stop it exiting the bone or the spleen. Okay, another option is, as I've said previously, is if you have a self-reactive B cell, so its B cell receptor responds against self-antigen, we have a last-minute chance to amend the immunoglobulin gene to make it non-reactive to self-antigen. If it's successful in amending the gene and making the B cell receptor no longer responsive against self-antigen, then that B cell can proceed to become an immature and subsequent mature B cell. Okay, so they're the options. Now, these are the options in a little bit more detail, I suppose. As you can see on the first scenario, the B cell the immature B cell undergoing the process of development doesn't react against self-antigen, and then can subsequently make its way and become a mature B cell and make its way to the lymph node. The next scenario is where the B cell receptors have the ability to bind to multiple antigens on a pathogen, causing cross-linking. And if this happens, then the B cell will undergo immediate clonal deletion, or the B cell receptor will be edited. Okay, so that's a scenario where the B cell receptors have the ability to bind to multiple targets on one antigen or one pathogen. The next scenario is where the B cell receptor binds to soluble antigen. Okay, it binds to very simple antigen. And when this happens, if it reacts against soluble self-antigen, it'll migrate to the periphery, but it will be programmed to become anergic. Again, this concept of anergy is where the B cell, even if it's stimulated by the self-antigen, will not result in an immune response. And if it comes into contact with a corresponding T cell, that T cell will kill that anergic that B cell. Okay. And then the next step is where it binds to soluble antigen, but the affinity is very low. And in this case, the B cell will migrate to the periphery. And because it has very low affinity, essentially that B cell will become clonally ignorant. So whilst it recognizes self-antigen, it just won't result in any strong degree of immune response. So you have full maturation, clonal deletion, receptor editing, anergy, and clonal ignorance. Okay, so there are the steps involved. So now that the immature B cell has now left the bone marrow, it makes its way towards the spleen. And when it makes its way towards the spleen, upon entering the spleen, it becomes a T1 cell, a transitionary 1B cell. Okay, and the transitionary 1B cell will undergo a check to make sure that it doesn't respond against self-antigen. Again, if the T1 cell does respond against self-antigen, it will be eliminated or amended. If there's no issues with self-reactivity with the T1 cell, it matures into a T2 cell where it can be checked again to see if it's self-reactive, but it'll also be checked to make sure that it can respond against the antigen that we want it to respond against. If it does respond against self-antigen, then it can differentiate into a series of mature B cells. Okay, it can mature into a marginal zone B cell, which is a MZ B cell, or it can mature or differentiate into a follicular B cell. Another B cell that it can mature into that's not indicated in the diagram is a B1 cell. Now, for the majority of the material that you'll be dealing with, you'll be looking at the activities of a follicular B cell. Okay? But for your own knowledge, a marginal zone B cell is a B cell that doesn't need T cell help, so it can become activated without the help of a T cell. And it also stays in the spleen. And the reason these B cells exist in the spleen is because it can sense or detect pathogens that are present in the blood. And because we have these marginal zone B cells in the spleen, it enables a very, very rapid response against that pathogen in the blood. Whereas the follicular B cell leaves the spleen, goes to the lymph node, and waits for antigen to bind to its B cell receptor, and then subsequently gets some help from T cells to elicit a response. And so that's the B cell that we'll be mainly focusing on for the majority of the module. Another B cell out of interest is the B1 cell. And this is another B cell that doesn't require T cell help. This is another B cell that can produce antibodies that actually have, this is a very unique scenario because most B cells produce antibodies or B cell receptors that are specific to one epitope or antigen. But these B cells actually have general specificity for multiple components, for instance, of a bacterial cell wall. So they help with a quick response to an infection on a very general level. Okay, so you can read up a little bit more about that in your own time, but just to highlight that the follicular B cell is the B cell that we're going to focus on for the majority of this module. Now, in terms of giving you some sort of idea of, how we've talked about negative selection in both the bone marrow and the spleen, and there is evidence that those stages also happen in other areas, such as the tonsils and that. So there is a certain amount of cells that manage at every stage to escape this negative selection. So you can see in the bone marrow here, the early immature cells, about 70% of them are autoreactive. And then when they go through the negative selection in the bone marrow, they manage to get that down to 40%. Okay? Now, the negative selection that happens in the bone marrow refers to a process called central tolerance, which we'll cover later in the lecture. Okay, so it goes from 70% autoreactive B cells to 40%. And then those 40% autoreactive T cells arrive at the spleen or in the periphery. And when they arrive at the spleen, 40% of these autoreactive B cells get eliminated, bringing that down to 20% of the B cells being autoreactive. Okay, so and that is this should highlight the difficulty in picking up these autoreactive B cells. Now, the interesting thing is that it's estimated that around 20% of B cells that then subsequently exit the spleen and travel to the peripheral lymph nodes, still about 20% of these B cells are autoreactive. And there's a lot of theories about why this might be. There's some theories that they may be useful in clearing cellular debris, but there's other theories that might result in increased risk of autoimmunity. Again, this is something we'll cover at a later stage. But this slide, in summary, is just to highlight the difficulty of picking up autoreactive B cells and the rationale for why we have multiple stages of negative selection. And of course, the negative selection that happens in the periphery, in the spleen, and other areas are referred to as peripheral tolerance, again, which is something we'll cover later in the lecture. Okay, folks, so this slide gives you a summary of how a B cell develops. It goes from a stem cell through a series of pre-B cells, where it rearranges its B cell receptor. We make sure that B cell receptor works. Also that we check to see if it reacts against self-antigen. At the end of that process in the bone marrow, it becomes an immature B cell. That immature B cell makes its way to the spleen. In the spleen, the B cell becomes a transitionary B cell, a T1 B cell. It goes through negative selection, differentiates into a T2 cell, transitionary 2 B cell, where again it gets checked, and it also gets checked to see if it has the ability to do its job, that is, to react against the antigen that it targets. And once that check is done, then if it's successful, it can become a marginal zone B cell which stays in the spleen, or it can become a follicular B cell which exits the spleen and makes its way towards the lymph node, where it waits for antigen and associated T cell help, and at that point then it will produce antibody. Okay? So, this slide summarizes the changes that happen in terms of the B cell receptor expressed. Okay, so when the B cell undergoes that process of gene rearrangement, it'll express the heavy chain with a surrogate light chain. And at the end of that process, when it becomes an immature B cell, it will express mature IgM. Okay? It then leaves the bone marrow, makes its way towards the spleen, and when it arrives at the spleen, at that stage it becomes a transitionary B cell where it expresses both IgM and IgD. And at the end of the process, when it becomes a naive, mature B cell, it expresses IgM and IgD. That mature B cell leaves the bone marrow, makes its way towards the lymph node, And when that B cell becomes activated in the lymph node, it downregulates IgD and expresses IgM. Okay? So be aware of those processes as well. Okay, folks, so that takes us to the end of the general B cell maturation process and the different forms of pre-B cell, immature B cell, transitionary B cell, and mature B cell the processes that it undergoes. Okay, folks, so that's a general summary. Now what we're going to focus on is the individual steps, okay? Firstly, we're going to deal with the processes involved in B cell receptor or immunoglobulin or antibody, whatever way you want to express it. We're going to give an overview of how that gene rearrangement occurs in the heavy chain and the light chain. Okay? And that process initially is called somatic recombination. Okay, so the general concept here is that when we get infected with a pathogen, the innate response will do a very quick and efficient job, but in many cases it's not sufficient to deal with the pathogen. So whilst the innate response has a really important role, it needs help from the adaptive response. It needs the general innate response needs another specific response to help it in its response to the pathogen. In order to develop this specific response, we need each B cell and T cell in our body to have its own unique receptors, okay? And in order to achieve this, each B or T cell, in order for them to achieve a unique receptor, it needs to go through a process of modifying its B cell receptor heavy chain and light chain. And it achieves that through a series of processes. The first is somatic recombination and associated junctional diversity. Okay, so somatic recombination, junctional diversity, and the last one is somatic hypermutation that takes place after the B cell becomes activated. Okay, so once it undergoes the initial somatic recombination and junctional diversity, this allows the B cell receptor, or if it's T cells developing, they undergo a similar process to develop a unique T cell receptor. So once it undergoes this process of rearrangement, those B or T cells will then have a unique receptor that will only recognize one pathogen. Okay? And this gene rearrangement enables our immune system to develop a specific response targeted against a specific pathogen. Okay, so for now we're going to focus on B cell receptor or immunoglobulin gene rearrangement. And this is the concept we have. There's a limitless number of microbes that we could be infected with from the environment. And the challenge we have is we need a specific B cell receptor or antibody for each individual antigen. Okay? Now, it would be impossible for our genome to have one gene for every individual pathogen that we have, which is why we have evolved to have a template B cell receptor gene or immunoglobulin gene. And what will happen then is each individual B cell will subsequently edit and modify that template gene in order to achieve their own version of that gene. If we wanted one gene for every pathogen, there would be no way that we'd be able to fit it in our genome. It would be too exhaustive. As I said, so our bodies have evolved this concept that we have a template heavy and light chain gene, and that we overcome this issue by using somatic recombination of the variable region of those genes and associated junctional diversity. And that process of somatic recombination, junctional diversity, and subsequent somatic hypermutation after activation enables us to achieve up to 10 by 10 to 11 different possible B-cell receptor possibilities from that one single gene. Okay? So, by the end of this process, you need to appreciate why the heavy and light chains need to be recombined. They need to be recombined because we need to ensure that each B cell has their own version of the gene. And in achieving this, you need to appreciate the 3 points listed in this slide. So you need to appreciate what somatic recombination is, which we'll deal with in a minute. And then you need to appreciate what junctional diversity is. And lastly, somatic hypermutation. So, let's start with somatic recombination. This is a scenario that we generally have. A B cell will have 2 copies of the heavy chain, 1 copy from the mother and 1 copy from the father. Okay? And it will also have 2 copies of the kappa light chain and 2 copies of the lambda light chain. Okay? So in summary, we have 2 heavy chains and 4 light chains. Okay? Now, we also have random nucleotides which will come into play later on at another stage. Okay? So that's what we have to appreciate. So what happens first is, so this slide is to highlight where these heavy chains are useful. So here in the green, you can see the two heavy chains. Okay, the green component of the heavy chain is the constant region. So that's a region that doesn't get edited or modified in this process of somatic recombination. The component of the heavy chain that gets modified is the variable region. Okay, and the variable region is composed of gene segments V, D, and J, okay, for the heavy chain. If we move to the light chain, it's a similar setup where you have the green component, which is the constant region. That's not modified in this process. But in the light chain, you only have 2 gene segments in the variable region, which are the V and the J, okay? So the heavy chain has V, D, and J in its variable region, and the light chain has V and J. Okay? And of course we have 2 heavy chains and 2 light chains which combine to make up the B cell receptor or immunoglobulin or antibody, whatever way you want to refer to it. So, at the start of somatic recombination, these are the steps that take place. It starts to recombine the heavy chain first. Now, we have 2 copies of that heavy chain. We have 1 copy from our father and 1 copy from our mother. So what the cell will decide to try and recombine one of these genes to start with. Okay? It also has 2 kappa chains, 1 from father, 1 from mother, 2 lambda chains, 1 from father, 1 from mother. Okay? Now, step 1 is the first step is to rearrange the heavy chain. Okay? So it selects one parent's gene copy first. So in this case, it wants to modify the father's copy of the heavy chain first. And here you can see there are a range of different possibilities. There's up to 65 V regions, up to 27 D regions, up to 6 J regions, and then of course you have the constant region of the heavy chain. And what happens basically is there's specific enzymes RAG1 and RAG2 that will help us edit this gene and cut out specific regions within this gene. So RAG1 and RAG2 start off the somatic recombination process by selecting a region of D and J. Okay, like this. Okay, so in this case, the cells decided to cut after D2 and cut after J4. Once they do that cutting, they cut out that segment of the gene, and that segment of the gene is then degraded and eliminated from the gene in this B cell. Okay, that's now gone forever. And what happens now is we have junctional diversity. And what junctional diversity is when we do this cutting out of the segments, at the end, there are sticky ends at the end of the D region and sticky ends at the end of the J region, and random nucleotides are added to these regions. Nucleotides can also be removed or added to these regions, and that is called junctional diversity. And one thing to appreciate is that when these nucleotides are removed or added, it's done in such a random way. Okay, so there's no process to decide how many nucleotides get added or what nucleotides get added. They just get added in a random fashion, and that's called junctional diversity. Okay, that they then can recombine. So now that gene has been edited permanently, and that is called D to J recombination. The next step is we want to edit the segment between D and J. The segment between D and J has now been completed. The next step is to recombine the segment between V and DJ. Okay, so RAG1 and RAG2 have their part to play again. They select a random region of the V. Okay, so they've selected just after V2, and they're going to also cut just after D1. They cut that region out, and that's then degraded and lost forever. And once again, junctional diversity takes place, where nucleotides are either removed or added between the sections and the whole gene segments recombine. At that point, the somatic recombination of the father's gene is complete. Okay? The final immunoglobulin heavy chain gene in the B cell has been amended. Okay? So then what happens is we need to check if this heavy chain rearrangement has worked. And the way it does this is it will transcribe the gene into messenger RNA and subsequently into a protein product. Okay, it'll splice out any unwanted regions and produce a heavy chain protein. And that heavy chain protein is then attached to a surrogate light chain. Okay, and the reason it's attached to a surrogate light chain is it just wants to test if the heavy chain has the ability to be functional. And what it does is it expresses the heavy chain with the surrogate light chain on the surface of the cell, and if that rearrangement is functional, it will get a signal to survive. Okay? And if it gets that signal to survive and the rearrangement has been successful, then what the B cell does is switches off the other copy of the gene. In this scenario, we do not need two copies of the heavy chain. So the fact that the first rearrangement worked means we don't need the other copy. And when you switch off the other copy, that's called allelic exclusion. Okay? We're now good to proceed. But the heavy chain's been rearranged successfully. But what if the heavy chain isn't successfully rearranged? Then what it can do is switch that gene off and try to rearrange the other copy of the heavy chain. In this case, the mother's copy of the heavy chain. And it undergoes the same process of D to J and then V to DJ rearrangement, junctional diversity. And if it rearranges that gene, checks it, attaches it to surrogate light chain, checks it to save it if the rearrangement has been successful. If the rearrangement has been successful, then the mother's copy of the gene will be used, and it will then subsequently proceed to light chain rearrangement. If that rearrangement was unsuccessful, it has an opportunity to re-edit that gene and to make it a successful heavy chain rearrangement. And if that's successful, then it can proceed to light chain. If after further editing of the gene it's unsuccessful, then because there was an inability to successfully rearrange the heavy chain in both copies of the gene, then the cell is instructed to die. Okay? So I'll try to edit both copies of the gene. If both copies are unsuccessful, then the cell has no choice but to kill itself, because there's no point in having a B cell with a heavy chain that does not work. Okay, folks, and in terms of then the series of steps involved, these are the steps involved in B cell rearrangement. Okay, first you have heavy chain rearrangement, which we've talked about, and then you undergo light chain rearrangement in a similar fashion. The only difference with light chain is you get Phi to J recombination, because there only is a Phi to J region in the light chain. And you'll start to, you will try to rearrange the first kappa gene. If that's successful, then the B cell receptor gene rearrangement of heavy and light chain has been successful, and you then have an immature B cell. But if the rearrangement of the first kappa gene was not successful, then you try to rearrange the second copy of the kappa gene. If that's not successful, you try to recombine the first lambda gene. If that's not successful, you try to copy the next copy of the lambda gene. If that's not successful, then the cell is instructed to die. Okay? So for light chain rearrangement, you get 4 opportunities to rearrange the gene, and if it's not successful at any of those stages, then the cell has to undergo cell death. Once you have undergone successful rearrangement of the heavy chain and successful rearrangement of the light chain, then the immature B cell will express IgM.